Here of the blood. This is a scanning electron microscope image that shows red blood cells in red, right? And then we have white blood cells in green, and then these smaller pieces or sacs of enzymes that are called platelets. So we're going to talk about these different formed elements of the blood as we go through talking about the circulatory system. So the first thing, I know this is anatomy that I like to express is some of the major functions, if you will, of the circulatory systems. Even though this is anatomy, remember form equals function. What the circulatory system looks like and its architecture will determine what it does functionally. Many of these, if you've had any anatomy at all or physiology, you've already heard before. So the circulatory system transports materials. It's what it is. It's our lifeline as vertebrates. All you have to do is look at the number or length of blood vessels that are found in the human body, for example, or any large vertebrate. That's a mammal. You as a human have about 60,000 kilometers of blood vessels running through your body. Most of those blood vessels are going to be capillaries, which are the smallest blood vessels. Most of that 60,000 kilometers are capillaries. Those capillaries are where exchange happens. And if you're billions of cells as a large vertebrate, and in mammals at least, no cell is usually more than 2 or 3 cells away from a capillary, then it starts to make sense that this transport system is extensive. And it transports blood, right? And blood is circulating and delivering material. So that's what the circulatory system does. It's a delivery system, if you will. And it also takes materials away from cells so that you can get rid of waste products. The big thing about cells, or most cells in the body, are attached. They're anchored. They can't move. If the environment deteriorates, they're going to die. And so the circulatory system is what keeps fresh materials coming to the cells and takes away waste products. So one major thing that you already know is the circulatory system transports gases, particularly carbon dioxide and oxygen. Oxygen's carried inside of blood cells, typically in vertebrates. A certain amount of carbon dioxide is also carried by blood cells, but most carbon dioxide, believe it or not, is carried as a different compound called bicarbonate within the plasma. It adjusts the changes in pressure, and I'll give you 2 examples of that later on. Seals, which dive, or whales that dive very deep, and also giraffes, which have to deal with their really tall heads coming down and up, for example, when they're drinking water. Transports excess heat. When you warm up, when you're exercising, you turn red, right? Your face flushes, your hands flush, your arms flush, you start sweating. You send all that warm blood to the surface so that you can use evaporative properties of water to dissipate heat from your body. It also absorbs heat. Cold-blooded animals like turtles, you'll see on a spring day when it's 60 degrees and it's sunny, you'll see them with their arms spread out and they're laying in the sun basking. They send their blood to the surface to gather heat to warm their bodies up. Transports all the nutrient molecules to cells from the digestive tract, things like glucose, amino acids, vitamins, minerals, fatty acids, nucleic acids. All those things are carried around by the circulatory system. Transports hormones and other chemical communication molecules which help coordinate physiology within the body. And also transports, as I mentioned before, waste products to organs that are responsible for recycling and detoxifying and dealing with waste, the kidneys and liver in particular. The blood also carries a number of cells, the white blood cells, which are responsible for immunity. And which makes sense. If you got 60,000 kilometers of places to hide, if you will, and you can get anywhere in the body, it makes sense that part of your defense system, your armory of cells, if you will, are going to be circulating in the circulatory system. So millions and millions of connecting tubes. Most of these tubes are the smallest blood vessels in the bodies, which we call capillaries. Okay, so this is kind of the typical vertebrate circulatory system. The big thing about the vertebrate circulatory system is it's a closed system. All right, and what that means are the blood vessels are all linked together. They don't open up into the body, and blood doesn't bathe the organs like it does in invertebrates, for example. So the blood is circulating through a closed system of blood vessels and doesn't drain out of the blood vessels to bathe the body. Therefore, we can keep it under pretty high pressure for the most part. And the major organ that's responsible for producing pressure is our pump, which is the heart. All right, so typically in a vertebrate, you have a centralized heart. Sometimes you'll have some accessory hearts that are found in other places, like we talked about earlier, right? That are responsible for helping return blood back to the heart. But our heart produces pressure by squeezing on the blood. Squeezing on the blood forces it into a closed tube. That tube will, the fluid that goes into the tube pushes on the walls and produces pressure. We call that hydrostatic pressure. That hydrostatic pressure we produce is called blood pressure. And blood pressure produces flow and causes the blood to circulate. Now, there are blood vessels leaving the heart, which are called arteries. All right, so we got, I don't have a red, but we'll do arteries. All right, so we have arteries in red. And then returning blood to the heart, we have veins. So those are the best definition of blood vessels. Veins bring blood back to the heart. Arteries take blood away from the heart. Most veins are carrying deoxygenated blood. Hemoglobin, which is the pigment that carries, the protein pigment that carries oxygen, is blue when it does not have oxygen attached to it and is red when it does have oxygen attached to it. And so veins typically carry deoxygenated blood. Not every vein carries deoxygenated blood, particularly in the pulmonary circuit or in the embryo. But most veins in our body and what we call systemic circulation are carrying deoxygenated blood. Most arteries are carrying oxygenated blood out to the body. But there are some arteries, again, in pulmonary circuit in the embryo that carry deoxygenated blood. Typically, we're going to have exchange happen only in the capillaries. Most of the really large blood vessels that you see in the body are simply just transporting tubes. They are transporting blood from one place to another. And when you get down into the organs, the blood vessels divide into smaller and smaller and smaller and smaller blood vessels until they become capillaries. And within the organs, those capillaries then deliver blood to the individual cells of the tissues. And so capillaries are where exchange happens. And what this figure shows you is where gas exchange happens. So where gas exchange happens between the environment and the blood is called external respiration. All right. So external and respiration. Respiration is the exchange of gases. And so the external respiration happens at gills, lungs, or the skin, for example, are places where you exchange gases between the blood and the environment. Down here where the tissues are, this is called internal respiration. All right, internal respiration is then where you exchange gases, oxygen for carbon dioxide, between the tissues in the blood. So there are kind of two circuits you can see here. There's a circuit of blood vessels up here. That's going to be called the pulmonary circuit, as you'll see. And then there's a circuit or a loop of blood vessels down here that we'll call the systemic circuit. And this would be a tetrapod because we have lungs, right? So fish only have a single loop, all right, what we call a single circulation. So we'll talk about those things next. So you can see that the blood is transporting materials to and from organs, delivering nutrients and oxygen and taking away carbon dioxide and waste. It also delivers things like hormones from the endocrine system. So remember, we talked about those a minute ago. So hormones are communication molecules, right, that tell certain cells to do something or change their physiology. We have the digestive tract where we pick up our nutrients, right, and get rid of our solid waste. We have kidneys where we filter the blood and actually clean the blood, which we've already talked about. And we have the liver, which is a detoxifying organ and an organ that stores nutrients that is attached to the digestive tract. And we got the heart pumping things everywhere here through the body. So that's a good kind of intro to circulation. And what we're pumping here is blood, right? So what is blood is kind of the next question. And that's what we'll talk about on this slide here. So blood is basically a liquid, right? And it's first and foremost considered a specialized connective tissue. All right, so that's what most histologists call it, me being a histologist. So it's a specialized connective tissue. And it's a connective tissue because the formed elements the actual solid parts of the blood are formed by bone marrow, right? So bone is part of the connective tissue. And so basically blood is broken down into two kind of parts because it's a suspension. Blood is a suspension. It's broken down into the liquid part, the watery part we call plasma, and then the part that would settle out if you put a drop of blood on a table, it settles out into a drop of kind of straw-colored liquid. That's the plasma, and then a clump of stuff that is solid, which are the formed elements. The formed elements are the blood cells. All right, and so we're going to have a slide on the blood cells, but there are three major types of formed elements. There are what we call erythrocytes, erythro meaning iron containing. That's what hemoglobin contains is iron. And those are the red blood cells. White blood cells are called leukocytes. Leuko means white. And there are a number of different types of white blood cells. They're involved in immunity. And then thrombocytes activate thrombin, right? That thrombin is the protein that they activate. And that's why they're called thrombocytes. Thrombin causes clotting, right? And so these cells are involved in blood clotting, platelets are. All of these are produced through this process here. We call it hemopoiesis. Hemo means blood, poiesis means the genesis or production of, and that's done within the bone marrow. All right, so that's the formed elements. The plasma is the watery part of the blood. It's 95% water and then has a bunch of things dissolved in it. Like proteins and other solutes, ions, communication molecules, sugars. It's what's called the ground substance or the matrix of the blood. All right, so it's what the blood cells are traveling in. And it typically is about 50% of the blood, and the formed elements are the other 50%, if you will. So that's what blood looks like. Let's talk about the individual formed elements. We'll start with red blood cells and then talk about platelets also here. So as you've seen in the lab that you're doing right now, blood cells, the red blood cells in particular, are very, very common, right? They're very, very common in the blood. They're actually the major cell made, and in mammals, we make a lot of them if you've done the counts, right? So the bone marrow is very busy. It's basically made up of stem cells. It's a stem tissue, and it has the ability to undergo high rates of mitosis, much like the integument, and it produces all of the cell lines that we call formed elements. And obviously there are millions of red blood cells, right, in mammals per microliter, right? And there are like 5 liters of blood that are traveling through circulation. So think of the number of red blood cells, right? That number is pretty much unfathomable in my mind. But what gives me kind of a general understanding of how many blood cells there actually are is in a mammal, typically 1 million red blood cells are being produced every second, and 1 million are leaving circulation every second to be recycled. That is how huge the number of red blood cells are that are being transported through mammals. So the typical red blood cell that all of you are probably familiar with is this one right here, right? Is this picture up here in the left top corner, or the one that's right here in the top right corner. These are the red blood cells of a mammal. And those are, you being a mammal, typically the blood cells that you see on the internet or in a textbook are mammalian red blood cells. They are anucleated. These cells during their development, once they mature in the bone marrow, right before they're released into the blood supply, they eject their nucleus. So they lack a nucleus. They have a few ribosomes and really lack any of the other organelles. And so they have a finite life because they cannot repair themselves if they're damaged because they lack the organelle machinery to do that. And so the typical red blood cell in a mammal lasts about 120 days of circulation. That's as long as it can last, and then it's typically removed from circulation and recycled. The blood cells are filled with the protein we call hemoglobin, all right? And you can see here is the protein hemoglobin. And what these cells do during their development is basically fill their cytoplasm with hemoglobin. In hemoglobin, its job is to carry oxygen and to drop oxygen off, right? So it's going to carry it in certain instances and drop it off in other instances. If you want to learn how it does this and why it does this, then you need to take physiology with me later on. But that's what red blood cells do. They carry oxygen. Almost all the oxygen in a mammal, in particular, is carried in the blood supply. And think about why that is. You're 98.6 degrees. And water at that high of a temperature carries very little oxygen. It has very little oxygen carrying capacity because it's very hot water. And oxygen isn't carried very well in hot water. So because that water is so warm in our bodies, we have to have a transport system. And that transport system is hemoglobin. Hemoglobin is contained in the cell because you can organize it and carry a lot more of it inside of the cell. And it also causes clotting. And so we can't just have hemoglobin floating around because it will form blood clots within the body. And so it's carried in a cell. And so you got basically red blood cells are sacks of hemoglobin, right? That carry around oxygen molecules. Now, all other vertebrates have nucleated red blood cells. So I like to at least show you pictures of those. So this is a reptilian capillary under an electron microscope that I took a long time ago. And you can see here are two red blood cells being transported in that reptilian capillary. And you can see that their red blood cells are nucleated. So they have a lot less cells, as do amphibians and fish, which you also saw with your blood count, right? And those cells are nucleated, which means they last a lot longer. They can last months, not, they can last 6-8 months, not 3 months or less than 3 months or 120 days, 4 months, I guess I should say. With mammalian red blood cells. They last a lot longer than mammalian cells do because they have a nucleus and the organelles that they need to actually repair themselves. They do also have mitochondria and the other organelles. So the reptilian red blood cells actually burn oxygen, which is not efficient, but they can be less efficient than us because they're cold-blooded and they don't necessarily have to carry the amounts of oxygen that we do. So they need less cells, and it's more productive on their part to produce cells that last longer because their metabolism is much slower. So the biconcavity of red blood cells, as you see here in mammalian cells, allow them to be bendable to actually fit through the capillaries because most of the red blood cells are about the size of the diameter of the capillaries, which is about 8 microns. So the typical mammalian red blood cell is about 8 microns in width. It's also biconcave to increase surface area, so it's easy to absorb and release oxygen. In this nice electron micrograph up here, you can see the actual cells, the epithelium, that lines all of the cardiovascular system, the entire cardiovascular system. And that epithelium is called endothelium. And endothelium is part of what we're going to call the tunica intima when we get to the blood vessels. And it is smooth and a simple squamous epithelium. And it lines all the blood vessels in the cavity, the chambers of the heart. It is involved with a lot of blood physiology, immunity, clotting, vasodilation, and control of how much blood can enter and leave capillaries. So it's a major player physiologically. Red blood cells are really thin. As you can see, they're about 2 to 0.4 microns in width, depending on where you're at. And they're about 8 micron, in thickness, and they're about 8 microns in width. The other thing that you can see traveling in the blood next to the red blood cells are these little sacs, or these little pieces of cells, which are called platelets. And this is an EM picture here of a platelet, as you can see, which is basically a sac of granules. Here's a blow up down here on the lower right of platelets, showing you all of the different granules that are found in their different sizes, different colors that are found within the platelets. And these granules are enzymes that are involved in what we call the clotting cascade. So they're involved with forming clots when blood vessels are broken and plugging those clots until the blood vessel can heal itself so that we can keep pressure high and keep blood cells within circulation. Okay, so that's a little bit about red blood cells and platelets. A clot looks like this. A clot forms when you activate some proteins traveling through the blood that are inactive. They're liver proteins. They are activated by the enzymes within the platelets, and they form a thrombus, which is a network of fibers that surround the platelets and form a platelet plug. That's what a thrombus is. And that would plug the spot where the vessel has been damaged or broken open. All right, so that is red blood cells and platelets. Let's talk about white blood cells now. So here are the white blood cells. So there's more diversity to the white blood cells than the red blood cells. So there are different cell types. Again, each one of these would have a line of development, if you will, from a stem cell within the bone marrow. And here are the four major cell types. There's another one that is usually a blue colored stained white blood cell that's called a basophil, right? And so we'll talk about that one. So there are going to be 5 main ones you need to know. Notice I call them fill and site cells. So you have eosinophils, neutrophils, and basophils. Those cells are named based on the staining properties of their granules. They're called, the fill cells are also called granulocytes. So a granulocyte simply means a cell, right, with granules in it. So the fill cells are granulocytes. They fill their cytoplasm with granules. Neutrophils have granules that are neutral in pH, and they're actually vacuoles and lysosomes that you see. Basophils have blue granules that are high in pH. That's why they stain blue. And the major protein in a basophil, since we don't have any information on it, we'll put it down here, is histamine. All right. And heparin. All right, that's an A. Heparin. These two cause thinning of the blood and vasodilation. So they are involved in inflammation and allergic responses. So basophils are activated when you have itchy eyes or runny nose, flushed face during an allergic response, or you get an area that has been cut and infected and it turns red and gets swollen on your finger, for example, gets really hot. That is a bunch of basophils in that area releasing histamine. And that would open up the capillaries and deliver blood to that area locally. And it's histamine and heparin are the two granules that are released by basophils. These are proteins that cause vasodilation. Eosinophils, they stain pink, as you can see. Their granules are pink, and they release chemical weapons. These granules are chemical weapons that defend against mostly parasitic worms. And so places like in the US, eosinophil count in your blood is really, really low because we have really good sanitation. You don't typically see a lot of eosinophils. They're actually hard to see in a blood smear. If you find somebody that has a very high eosinophil count, or if you're a vet that finds a dog that has high eosinophil count, then you have a parasitic worm, like a heartworm, for example, or some other parasitic worm that's traveling through circulation. And so these chemicals are really like missiles. They blow holes in the worms and the worms explode. That's pretty neat. That's what eosinophils do. So those are the fill cells. The other two cell types are called monocyte, or called site cells. One's a monocyte and the other one is our lymphocytes. Excuse me. Okay, monocytes are the largest of the white blood cells. Monocytes travel through circulation when they're created, and then they leave circulation typically and enter the interstitial fluids or the fluid or areas around cells within tissues, and they differentiate into macrophages. So monocytes become macrophages, and the macrophages are going to protect the body by doing phagocytosis mostly. They can release some chemical weapons, but mostly they would run into something foreign. They would grab onto it, ingest it, pull it into the cell, and break it down. And that's what monocytes do. So they are going to be able to recognize our cells from foreign cells and ingest those invaders and rid the body of them. So those are phagocytic cells. The lymphocytes, there are two types. There are actually several types of lymphocytes, but the two major types are A and B lymphocytes. The A-type lymphocytes are going to also be similar to macrophages. They're going to be a phagocytic cell, a cell that goes out and actually makes contact with foreign invaders and ingests them and destroys them. The B cells, they actually typically stay in the immune system tissues like lymphatic nodes or lymph nodules or somewhere out in the body where they kind of are stationed. And the B-type lymphocytes, they differentiate when they're activated into plasma cells. And these plasma cells produce antibodies. So they can be found out in connective tissues, particularly areolar connective tissue, or be found in an immunological organ like the thymus or within lymph nodes, for example. And they can proliferate and produce antibodies that are specific to an invader. So what you're activating when you use, for example, a vaccine, or if you're then exposed to the flu after the vaccine, is you're activating the memory of that flu virus, and you will produce antibodies from these plasma cells once the flu infects you that will send those antibodies out and destroy the flu viruses before they can cause sickness. And that's how, for example, the COVID vaccine works. And the response you get, right, from the COVID vaccine are from activation of these B-type lymphocytes into plasma cells that produce antibodies for COVID. And that they use antibody tests to see if the vaccine's effective or to see if you've had COVID, right? And that's how the plasma cells work. Now, you can divide these cell types into phagocytic cells versus chemical producing cells. So your two major phagocytic cells are neutrophils and monocytes, and then some of the lymphocytes, the type A lymphocytes, for example. Neutrophils are actually the most common white blood cell that you'll find in a blood smear. They make up about 70% of your white blood cell population. And they go out and are just generalists. They will destroy anything that they recognize as foreign. They are typically the first white blood cell to reach a site of damage in a blood vessel, and they enter the tissue and eat bacteria or any invader that might get in through the broken skin, for example, and also clean up the area of debris and form what is called pus often that you see when you break open a wound or you pop a pimple. That pus are neutrophils that have died in defense of your body. So that's an introduction to the white blood cells. You should, for the exam, know kind of the names of each one and kind of their general function within the body. You can see up here in this blood smear here, we have two white blood cells. This first one with a lot more cytoplasm around it, and which is larger than all the other cells. This is a monocyte. This one right here is a lymphocyte. It has less cytoplasm. It's a little smaller. It's either a B or an A type lymphocyte. All right, so that's the blood cells. Now we can start talking generally about single and double circulation and kind of how the blood vessels are organized. Now, I'm not going to teach you every single blood vessel and what it does. You kind of get introduced to that in lab, right, where you're IDing blood vessels. Really, I want you to know the general themes and patterns of circulation as far as blood vessels go. We have a centralized pump, our heart, right? We're going to talk about kind of heart anatomy a little bit, and then we'll also talk about blood vessels, right? So there are two general patterns that exist in the vertebrates. They're called single circulation and double circulation, and this has to do with the number of loops that are attached to the heart. So single circulation is the primitive or basal condition, the ancestral condition. It's found in fish, and blood only passes once through the heart. So blood comes into the heart and leaves the heart in just in one direction. That means that the respiratory organ is basically in series or within the loop. And so here is a picture of what single circulation looks like from a fish. This is from your textbook, showing you we have our heart, and the heart, all the systemic capillaries, and then the gill capillaries are all in the same loop. That's called, they're all in series with each other in the same loop or circuit. So the heart produces pressure that it forces blood out to systemic circulation. These are going to be arteries that connect the heart to systemic circulation. And then the veins collect the blood from systemic capillaries and then collect it into large veins and deliver it to the gills. The gills then drain into the heart, and then the heart pumps that oxygenated blood out. So it's found in fish. Double circulation is found in sarcopterygian fish and also all tetrapods. And so double circulation involves two loops. So there is the systemic loop, and then there's the pulmonary loop. And basically the heart is going to go from a 2-chambered system to a 3 to 4-chambered system. And you have two upper chambers that are called atria that can receive the blood from that second loop. And so this loop that is called the pulmonary loop, it connects the heart to the lungs. And then the systemic loop connects the heart to all of the other capillary beds. So as I mentioned, external respiration happens between the blood supply, the capillaries, and the environment, and can include lungs, gills, or the skin. Internal respiration happens as systemic circulation. It's the systemic capillaries that are involved with exchanging oxygen and carbon dioxide between the blood and the tissues. Arteries, again, carry blood away from the heart. Veins carry blood towards the heart. And then you can have one loop or you can have two loops, right? And we're going to kind of talk about how they look in the different vertebrate groups. Amphibians and reptiles show intermediate patterns in their hearts and in circulation. Even though they look intermediate, I want you to remember that they're not directly linked evolutionarily to the ancestral and the derived conditions. They are under their own independent evolutionary trajectories, and even though they look intermediate, their structure fits their niche perfectly, the niche that they've evolved to take advantage of. Amphibians, for example, are amphibious. Part of their life cycle is aquatic, part of their life cycle is terrestrial. Adults can breathe underwater, and many of them can, amphibians, and breathe in air, which is a great evolutionary adaptation to escape predation and to also feed. So we'll talk about that. So let's look at some of these features. If I were to throw circulatory circuits or patterns up on a phylogenetic tree, basically you can see that the basal condition down here is the single loop, right? And then the derived conditions up here are completely separated double loops, where the heart is basically two separate pumps. One side of the heart receives blood from the pulmonary circuit, and one side of the heart receives blood from the systemic circuit. So you have two separate pumps, if you will, in the mammalian and avian systems, for example. So you can see up here, the most, what we consider derived system as far as circulation. And then down here, the fishes have the most simple system as far as just one loop with the gills and the heart and body, systemic capillaries, and series. Then you got some intermediates here, right? You got sarcopterygian fish, where you start to develop the first gas bladders that are attached to the digestive tract or to the exterior, and you get the development of the first three-chambered heart, as you'll see. And then the amphibians, which look similar to this, that have the lungs and the heart in the pulmonary circuit, but notice we also add the skin. The skin is really, really important in amphibians as a respiratory organ, particularly in salamanders. And then you got your crocodilians and your reptiles here, which have three-chambered hearts, and so blood can mix within them, but they do have the two separate circuits. And then the bird and mammal have these separate circuits separated in the heart, so bloods can't mix. So these intermediates look like really nice intermediates that show the evolutionary trajectory of, or at least appear to show the evolutionary trajectory, right, of circulatory patterns. Again, I want you to take caution in that. Remember, evolution doesn't work in a step ladder form up towards mammals. Each one of these lines have undergone their own independent evolution over millions of years, and the circulatory pattern you see fits the niche that these animals occupy. Just to show you that, I want you to know that double circulation obviously has arisen twice in vertebrates because birds and mammals aren't closely related to each other, right? Birds are more closely related to reptiles, which have the three-chambered heart instead of the four-chambered heart. And the complete double circulation has at least risen two independent times through convergence. And so there's no direct ancestry in those two groups. There are some weird specialized breathing patterns I want you to notice down here, and I can mention several of these. Some fish, some specialized teleosts actually can use the digestive tract, the intestines, to actually extract oxygen, and which makes a lot of sense because the intestines are heavily vascularized because they are absorbing nutrients, right, from what we eat. There are catfish, for example, that can be thrown out of the water and they gulp air and swallow it into the intestines, and they extract gases through the intestines and can survive out of water for long periods of time. So I like this image here. This image kind of shows your kind of three major patterns. Here's your single loop. Here's your double loop with your two separated pumps, if you will, of the heart. So notice the deoxygenated blood never mixes with the oxygenated blood on the left side of the heart. So the right and left sides are completely separated as blood enters the heart through a septum. The intermediate pattern, you actually have bloods that could mix because oxygenated and deoxygenated blood are sharing the same ventricle. I will talk about that really doesn't happen in the amphibians or the reptiles, and kind of how the heart can be three-chambered. Look, anatomically, that is, it looks three-chambered, but functions as a four-chambered heart. We do have the two circuits, right, in the intermediate pattern and in the derived pattern. So you have the pulmonary circuit and the systemic circuit. These circuits here, the pulmocutaneous circuit, which involves the lungs, pulmocutaneous, and the skin, and then just the lungs in reptiles, birds, and mammals, notice have evolved so that these animals can be effective tetrapods. They're not using gills, if you will, like fish. Again, we talked about the danger of saying these intermediates evolved the double circulatory pattern, right? There's a step-like effect as far as evolution goes. We got to be careful of that because each of these trajectories have evolved independently from one another for millions of years, and they fit the niche perfectly. And basically this mixed intermediate system here that you see with the heart allows amphibians in particular and a lot of reptiles to shunt blood away from the lungs. And to do that, you need the blood coming in from the right side and left sides of the heart to actually share a common chamber, the ventricle, so that you can shunt blood away from the heart. And I'll kind of explain how that works and show you some examples of how that works in the different intermediate groups here. And that design, being able to shunt blood from the lungs when they hold their breath underwater, allows them to stay underwater for a lot longer period of time, and in some cases even breathe effectively underwater. So each design, the intermediate design, the ancestral design, and the derived design, are all perfect fits for the niches that these animals occupy. This slide here shows mammal dual circulation. And as you can see here, if you follow the blood flow, the right side of the heart in blue, it's receiving deoxygenated blood from systemic circulation. So all of the capillary beds out in the body are going to drain into veins, all right? And these veins are going to merge into larger and larger and larger veins until you get to the big veins, the systemic veins here, which the biggest ones are called vena cavas, all right? And there are typically two vena cavas. There's a cephalic one and a caudal one, or what they call superior and anterior vena cava. And these large vena cavas, they drain all of the blood back to the right side of the heart. And that blood will drain into the right atrium. The right atrium is connected to the right ventricle through a valve. And there's a septum that separates the right side of the heart from the left side. So in reality, you have two separate pumps. You have a right pump and a left pump. The right pump here, once the blood enters the atrium, is going to be passed through this valve here called the atrioventricular valve, the right atrioventricular valve, into the ventricle. The ventricle then will contract and force blood up into the pulmonary circuit. This is called the pulmonary trunk. This is called the pulmonary artery here. The pulmonary artery is the only artery in the adult tetrapod that carries deoxygenated blood because it's carrying blood away from the heart, right? The pulmonary artery will then carry blood to the lungs, where it breaks down into pulmonary capillaries, which pick up oxygen and drop carbon dioxide. The oxygenated blood is collected by the pulmonary veins and delivered back to the heart, left side of the heart, via the pulmonary vein, which drains into the left atrium. The left atrium is connected to the left ventricle via a valve here called the left atrioventricular valve. And when the ventricle receives that blood and contracts, it forces blood up into the largest of the systemic arteries, which is called the aorta, right? So the aorta then is going to break down into smaller and smaller and smaller arteries and deliver blood to all of the smallest arteries in the body, which break down into capillaries, which then feed all of the organs and deliver oxygen and collect carbon dioxide. So external respiration up here, right? So external respiration up here and internal respiration down here, right? All right, just so that we're connecting the kind of the material we talked about previously. Okay, our next slide will then talk about what are aortic arches, and I'm going to save that for the next lecture, but the aortic arches are basically what connect the heart in fish to the gills. And then we're going to talk about how those change over the evolutionary trajectory of tetrapods, because they have to form other blood vessels within the tetrapods that are associated with the lungs. And so that's where we're going to go next. And I will do that in the next lecture. Have a great day.